All right, so in the last video, we ended the video talking about semen production, but I want to go ahead and review this section again before we finish the rest of the PowerPoint. So semen is also called seminal fluid. This is the fluid that is ejaculated during an orgasm. The semen is consistent of sperm as well as the fluid that the sperm is bathed in. So during ejaculation, a male will ejaculate usually between 2 to 5 mils of fluid. Now, to put that in perspective, a teaspoon is 5 mils. All right, a teaspoon is 5 mils. So a person can ejaculate as little as half a teaspoon and up to a full teaspoon with each ejaculation. That ejaculate material is made up of 60% fluid from the seminal vesicles, 30% fluid from the prostate, and 10% of everything else, which is your sperm, as well as the fluid from your spermatic ducts. For a male to be considered healthy, fertile, with a normal sperm count, there must be 50 to 120 million sperm per milliliter. So if they have 5 mils of ejaculate or a teaspoon of ejaculate, there should be 5 times these numbers. So that would be 250 to over 600 million sperm per ejaculation. To be considered infertile, there needs to be less than 20 to 25 million sperm per milliliter of ejaculate, meaning if they ejaculate a teaspoon of semen, there will be, 25, so about 125 million sperm per milliliter, per teaspoon of ejaculate, which is far less than what we see in the normal ejaculate material. All right, so those numbers are important. This is how we tell if a male is fertile or infertile. Now, sidebar. It is noted that it seems like male sperm count overall is starting to decline, and that could be due to several different things, including changes in diet, decline of health of the community, and of just males in general in their various communities. It could have to do with men wearing tighter pants. So remember, a few years ago, all the rage was skinny jeans. I'm sure it's going to come back sometime soon, but think about that. If a sperm cell the temperature needs to be regulated. And so you have all of these mechanisms for the scrotum to come closer to the body or further away from the body, depending on how much heat they need in order to regulate the heat so it doesn't overheat and decrease sperm count. When a guy wears skinny jeans or even tighty-whitey underwear too much, too long, then the scrotum, the testes will sit too close to the body too often, and that can decrease fertility. All right, so all of those things have a factor in how fertile someone is. So, as mentioned before, the prostate produces 30% of the ejaculate fluid. That prostatic fluid is a thin, milky white fluid. It has things like calcium, citrate, and phosphate ions in it. It also has a clotting enzyme, and there is a protein hydrolyzing enzyme. So This is an enzyme that hydrolyzes, obviously, a type of protein. And this is your serine protease, also known as prostate-specific antigen, or PSA. Your seminal vesicles produce this part of this 10%, I'm sorry, part of this 60% of your prostatic fluid. The seminal viscous fluid is a viscous yellow, yellowish fluid. Viscous means thick. It doesn't flow as easy as water because it is thick or thicker. This fluid contains the fructose and other carbohydrates. These are the nutrients or sugar sources that the sperm will use in its journey from the male inside the female, and hopefully it will be able to reach the egg, but it needs to have nourishment along the way. And so that is where it gets the primary portion of its nourishment. There's also citrate, there are prostaglandins, and there's a protein called seminogelin. Remember again, anytime you have pro in front of a word, that means that it is an inactive form and something happens. There will be an enzyme that will cleave this pro part off to make it an active molecule. So semen tends to be more sticky. And the reason why it's more sticky is because that helps promote fertilization. So you have this clotting enzyme in the that comes from the prostate fluid that activates that seminogelin that converts it to the active form of seminogelin. What it does is it entangles the sperm. It forms a bit of a loose, sticky clot or so that will stick to the walls of the vagina and the cervix. And this helps the sperm stay in the vagina, because remember, as soon as she gets up, gravity is going to be pulling anything that's not attached to the body. It's going to pull it off or pull it down, right? Pull it down with the force of gravity. So instead of having all of the sperm that he just ejaculated leak out of the vaginal vault, it will stick so that it can stay, and the sperm have a greater chance of being able to swim up into the or through the cervix into the uterus and hopefully meet up with an egg. So again, promotes the uptake of these clots into the uterus. After the ejaculation, so 20 to 30 minutes afterwards, then you will have this serine protease that will break down the seminogelin and cause it to be liquefied, so that now the sperm will have an easier time to swim and go to where it needs to go and hopefully meet up with an egg, and might possibly be the winner that will fertilize the egg. In order for sperm to be motile, there needs to be two different things. There needs to be an elevated pH, so you need a higher pH level, and you need an energy source. Remember in class, we mentioned about the pH of different parts of the body. So everything has an optimal pH. You have your stomach pH that is closer to 2, which is very acidic, and that acidity is necessary to turn on your some of your gastric enzymes to make them more active and to function better. If there was a higher pH in the stomach, those enzymes wouldn't be turned on, and so you would not be able to digest as well. This is, again, sidebar, this is part of the problem when a person takes things like Tums, or maybe drinking alkaline water or taking baking soda. You change the pH, you raise the pH of the hydrochloric acid in the stomach, making it less able to activate the digestive enzymes. Same thing goes for the the acidity of the vaginal vault. When you have the vagina is normally more acidic, so the sperm cannot live or cannot function optimally in that lower acidity or in that lower pH. So pH was normally around 3.5. Sperm do not do well in that pH. So part of the prostatic fluid will increase the vaginal pH to about 7.5. And then you have the fluid from the seminal vesicles that provide your fructose, that provide your other sugars that the mitochondria will use to actually produce ATP, so that that ATP will be that energy source for the sperm cells. The active sperm then crawls or swims up the vagina through the cervix into the uterus, and the prostaglandins in the semen will thin the mucus that is naturally found in the cervix, as well as in other areas of the vagina's uterine area, so that it's easier for the sperm to get to where it needs to go. And those prostaglandins may also stimulate contractions in the uterus to help the sperm get to where it needs to go to try to fertilize the egg. All right, so now let's talk about some anatomical foundations. So in this slide, we're basically going to talk about blood flow to the penis and from the penis. In order for there to be an not an ejaculation, but in order for there to be an erection, there has to be an increased blood flow to the area. That is what's causing the erection, is increased blood flow to the tissues of the penis that will engorge the penis with blood and will make it stiffer, right? And so that is how the male is able to perform sexual acts, because the penis is full of blood, right? And so you have this artery that will help to increase that blood flow when it's necessary. So the internal pudendal artery or the internal penile artery will enter into the root or the base of the penis, and then it divides into two other arteries. You have your dorsal artery, and then you have your deep artery. The dorsal artery is located, you should be able to guess, on the dorsal surface of the penis beneath the skin. This supplies blood to your skin, to the skin of the penis, to the fascia, and to a tissue called the corpus spongiosum. And I'll upload some images so that you can actually see what we're talking about. So again, dorsal artery will supply blood to the skin, fascia, and corpus spongiosum. When the penis is flaccid, when it is not erect, most of the blood in the penis, because it still has to have a blood supply in order to live, still needs oxygen and nutrients in order for that tissue to live. So there is blood flowing, it's just not engorged, which is why it's flaccid and not erect. Most of that blood will come from this dorsal artery. Now, the deep artery will travel through the core of the corpus cavernosum. So there is the corpus spongiosum that makes up the penis tissue, and then there is the corpus cavernosa that makes up part of the penis tissue as well. You have the deep artery that travels through the core of the corpus cavernosum. This will give off smaller arteries that will penetrate the trabeculae. So I want you to look up the word trabeculae so that you can know what it is. FYI, this is I think this is the plural form of the word. But anyway, look up the word so that you can know what it means. It penetrates the trabeculae and then enters the lacuna. Make sure you're looking up lacuna. And this will cause a dilation of the deep artery, or the dilation of the deep artery will cause the filling of these lacunae, and this is what will cause an erection. All right. There are lots of anastomoses that unite both of these artery. And again, look up the term anastomoses, but then this is basically where you have various vessels that kind of fuse together. All right, so look up the term so that you can understand it better. Then you have the deep dorsal vein. Deep dorsal vein. This is what drains the blood from the penis. So after erection, after ejaculation, when the penis goes flaccid, it's because the blood from this deep artery in the corpus cavernosum, it will go back into the deep dorsal vein to leave the penis and re-enter the regular blood flow. So erectile dysfunction, again, erectile dysfunction is the inability to have an erection or maintain an erection sufficient for sex. In order to treat it, there are ways that you can treat it. Mostly there are drugs like Viagra, Levitra, and Cialis that are used. Now you should have been exposed to this through your pharmacology class by now. If not, just know that they're coming. But these are phosphodiesterase inhibitors. Phosphodiesterase inhibitors work in this area here. So it starts off where you have a sexual stimulation. So he thinks about something, he sees something, he smells something, he's rubbed, he's touched. Whatever that stimulation is for him, the stimulation will occur. That will cause nitric oxide to be produced. Nitric oxide will increase blood flow by turning this guanylate cyclase from an inactive to an active form. This will cause GTP to be formed to CGMP. And the rest of the, and then here you have this phosphodiesterase that will allow for the loss of an erection. So when you are ready to finish the erection, so let me back up. So cyclic GMP will continue and cause the vasodilation and erection. Once it is time for the erection to be over, then phosphodiesterase will actually change this CGMP to degrade it, and that will cause the loss of erection. Apologies for my stumbling. But that's basically what happens from erection to loss of erection. Phosphodiesterase will act. So these medications will act as inhibitors so that this enzyme doesn't convert CGMP or doesn't degrade CGMP, right? Which allows an increase in the amount of CGMP, which will allow him to have an erection and have it for a longer period of time. Longer period of time than he would if he were just naturally going from erect to not erect. And this is, of course, the desired state, the erection. Now let's talk about these sexually transmitted diseases, or more recently called sexually transmitted infections. Also, you'll see STIs more than you'll see STDs these days. So just keep that in mind. But many STDs will have an incubation period, which will be that period of time where the pathogen will start to multiply, but the person will have no symptoms. So this is the time where a person can be able to spread disease or spread infection, but they won't have symptoms because there's not enough pathogen to actually cause symptoms yet. All right, so this is communicable diseases where the person may not have any symptoms at that point. There are also people who are carriers but have no symptoms at all, even later on after the incubation period. These are symptomless carriers. So just to make this concept more relevant, this is part of the discussion about masks or no masks when you're talking about COVID and COVID infections. because you may have these symptomless carriers, people who actually have the virus in their bodies, but they don't have symptoms, they don't feel ill in any way, but the virus is actively in the body. So this concept of symptomless carriers exists not only in STDs, but also in COVID and many, many other infectious processes where the contagion, whether it's a bacteria or a virus, is there, it's growing, it's active, but the person who it's in does not have symptoms. So that is a pretty, I want to say dangerous. So dangerous is more harsher word than I want to use, but that's the concept. This is a pretty dangerous period because this person can be spreading disease to anyone they come in contact with. So you have your bacterial STDs or STIs, excuse me, and you have viral STIs or STDs. So your bacterial STDs include chlamydia, gonorrhea, and syphilis. Chlamydia may cause a urethral discharge and testicular pain. So if he's walking around and has a discharge from the penis, or he has pain in his testicles, that could be an indication that he has chlamydia. Again, he may not have any symptoms at all and still be a carrier of the bacteria that causes chlamydia. Gonorrhea will also have a discharge. This will be more of a pus type of discharge along with pain. And this one can actually result in sterility because of the inflammation of the pelvic area, or what's called pelvic inflammatory disease, right? So gonorrhea can lead to infertility by pelvic inflammatory disease. Syphilis causes these hard lesions wherever the site of infection is. These hard lesions are called chancres, or I guess that's how it's pronounced, chancres. So syphilis caused these hard lesions. At the first stage, after the first stage, the chancres will disappear, and then there will be this pink rash. That is the second stage of syphilis. And then the third stage of syphilis is called neurosyphilis. This is where you have cardiovascular damage and brain lesions. Now, if you remember the Tuskegee experiments, the Tuskegee experiments weren't necessarily about giving the men who have syphilis I don't think they actually gave syphilis to the men. What they did was they withheld treatment from the men to see what it would do if syphilis lasted a long period of time without treatment. Then this is part of the reason they figured out all of these different stages of syphilis. So again, Tuskegee experiment wasn't necessarily that they gave syphilis to people, which they may have done, but the biggest portion of it was that they knew these men had syphilis and they withheld the syphilis treatment from them to study what happens over a period of time if a person is infected with syphilis and not treated. And they found that there are 3 stages. So you have the first stage, which is the hard chancres at the site of infection. Once those disappear, you go into the second stage where you have this widespread pink rash. And then you have the third stage that affects your cardiovascular system and your nervous system. This is neurosyphilis. So know these. And then your viral STDs include genital herpes, genital warts, hepatitis B, and hepatitis C. Genital herpes is the most common STD in the United States. There are blisters and there are pain, and these blisters and pain will come and go. You almost never really get rid of a virus, especially a herpes virus. It just goes dormant and active, and this is why people will have outbreaks. You also have genital warts. So genital warts are warts that are located on that perineal region. which is, again, that space on the external anatomy between the rectum and the anterior sex organs on male and female. You may also have warts on the cervix, so inside the body, as well as um, warts on the anus. So depending on where the sexual act occurred and where the virus particles set in, you can have warts, genital warts on any of those locations. Hepatitis, remember, hepa means liver, itis means inflammation. So hepatitis B and C are viruses that will attack the liver and cause inflammation of the liver. All right, now let's look at some of the images of the things that we've been talking about. Remember that you do have your book. This is not going to be all the images, and it's not going to be as inclusive as your book. So utilize the book for the images so that the concepts that we talked about can become more real and you can actually see what we're talking about, see how these things look, how they function, where they're located, etc. So first image is just an image of sperm cells that have gone from the vaginal vault through the cervix into the uterus. They've gone through the mucus in the cervix. They've gone into the uterus, traveled up the uterus and into the fallopian tubes, because as we'll find out when we talk about the female reproductive system, that's where the fertilization of the egg by the sperm occurs, in the female fallopian tubes. So early in the chapter, one of the things that we talked about was how there is genetic variation in the species, because as the species continues, the offspring will have genetic mixing. So let's say that this blue is from mom, the green is from dad, and this is your genes, right? Half from mom, half from dad. Well, what basically happens is that there is a cross somewhere along each of these chromosomes that will allow for the mixing of these chromosomes. So now you have some of this from dad that is on mom's chromosomes, and vice versa. You have a little bit from mom that's on dad's chromosomes. And therefore, once it's time for you to share your genes with your offspring, it's not all dad or all mom, you have a little bit of both, and that creates the variation of the human species, so that your children will be somewhat like you, somewhat like your parents, but they will be their own individual people. This is part of the reason why that happens. And this further shows you how you have different crosses, different crossovers between your genes. And then you have that further differentiation, or you have the further breakdown where you have not both of these halves of the chromosome, but one half goes here, one half goes there. And this will be the sperm cell or the egg cell, where you have just that half of the genetic material, and then it will mix with the other to produce a human that has a full amount of genetic material. So here is the male anatomy of the reproductive system. So you'll see here that you have the abdomen, you have the pelvis, and then you have the outside of the body where you have the external genitalia. So just to orient yourself, these are your large intestines, right? So this is more than likely your transverse colon. This is the descending colon, and I know it's the descending colon because here's the rectum and the anus, and that is the end of your descending colon. You also have, so I'm starting with the organs that you already know. You also have the ureters. Remember, there are two ureters that feed into the bladder. All right. I want you to pay attention to the location of this. This is on the posterior side of the bladder. So if you ever see a model or an image of a bladder and you can actually see where the ureters connect to that bladder to fill the bladder, then you know that that is the posterior side of the bladder, because on the anterior side, there are no ureters. You also have from the bladder, the urethra that goes through the prostate and then out through the penis and then out of the body. This is your pubic bone. So remember the ramus of the pubic. It's cut so that you can actually see half of the body, of course, but that is what this is. So that is the anterior side, because that is where the pubic, this part of the pubic bone is. You have the pubic symphysis somewhere up here, is probably closer to that part, but this is what this is. So that's anterior, this is posterior. You have your prostate here. Remember that your prostate produces the prostatic fluid. You also have your seminal vesicles that produces some of the fluid for the ejaculate. Both of those will add material into the, add material and sperm into the urethra for ejaculation. You have your testes here that will produce the sperm. You have the epididymis where the sperm basically kind of sits and hangs out as they're maturing. And then when it's time, when they're called, they're ready to go, and they travel from the epididymis through the vas deferens or the ductus deferens. Both of them are the name of the same tube. That will travel on the superior side of the bladder and then wrap around the posterior side of the bladder, and then will open up into the ejaculatory duct, same as the seminal vesicle is opening up into the ejaculatory duct, and they will ejaculate or put their fluids into the ejaculate, and it'll go from that ejaculatory duct into the urethra and eventually out of the body. You have the bulbourethral gland that also produces some of the part of the ejaculate that sits here inferior to the prostate, that will also ejaculate part of its, or incorporate part of its fluid into the ejaculate. Penis is here. Your penis is made up of your glans penis, which is the head of the penis, the prepuce, which is the foreskin or part of the foreskin. You have your corpus cavernosum here. You have your corpus spongiosum here. And these are the tissues that when they become engorged with blood, they make the penis more full and stiff, right? So that is what ejaculation is the filling of the penis with blood, the filling of these spaces with blood, this corpus cavernosum and corpus spongiosum. Let's see, what else can we point out? That's basically it. Yeah, except the scrotum. So remember, the scrotum is the sac of skin that the testes are housed in. All right. So this is when you have, when he's cold and the testes move closer to the body, the muscles will tighten and will wrinkle the skin of the scrotum. And that's because the skin doesn't tighten. The skin is not muscle. It's only epithelial tissue, well, epithelial tissue and connective tissue. So it's not going to tighten or anything. And that's why it gets wrinkled when the testes are pulled closer to the body for extra warmth. So here's another view. Hopefully by now you know that this is a posterior view of the organs because you can see this ureters empty into the bladder, but you also know that it's the posterior side because these seminal vesicles are on the posterior side. Let me go back to the previous image. The seminal vesicles are here. I don't know why that got so big. Not really sure what happened. But these seminal vesicles are located on the posterior side of the bladder. So if you ever see seminal vesicles or ureter or the ampulla of Vater, which is the tube where we'll get to it. Seminal vesicles. I don't know why that's doing that. Seminal vesicles are posterior side of the bladder, and that will empty into the urethra. All right. You have the testes. The epididymis is where the sperm will go, and basically, once they're created, they kind of sit there to mature, and then when they're ready, they will go through the ductus deferens or the vas deferens into the ampulla of Vater, and then they will join the ejaculate. Bulbourethral gland is just deep or inferior to the prostate gland. All of them have an opening into the urethra, and then it can go out of the body, right? So you have the penis, the shaft of the penis or the body of the penis is here. The root of the penis is closer to the body. So the portion of the penis that's closer to the body is called the root of the penis. In the root of the penis, you have the bulb of the penis, which is here, and the crus of the penis that is there. You have the glans, which is the head of the penis. And that's it. So make sure you are studying this. Please, please, please make sure that you are looking at your book so that you can get an even better orientation of what all of these are and where they're located, what they do. When a child or a male child is younger, as I mentioned before, the testes start off in the abdomen. So you have the testes that are higher up. This is your abdominal pelvic cavity. So remember, you have the membrane that lines the cavity. And if you go back to the urinary system, remember we talked about the kidneys as being so this is the peritoneal cavity. The kidneys are located in the retroperitoneal space, retro meaning back or throwback. So the retroperitoneal space will be the space behind this peritoneal membrane, retroperitoneal. So according to this picture, the testes are also in a sort of a retroperitoneal type space because it's not actually in the abdominal pelvic cavity because of that membrane that separates it. So testes start higher in this retroperitoneal cavity. Remember, you have this gubernaculum, which is a ligament that attaches to the testes as well as the pubic area, the internal pubic area. And so as the testes are getting ready to descend into the pelvic cavity and eventually outside of the body, this gubernaculum will guide it down. All right, this is the developing penis. So this is a fetus, right? An image of a fetus, just to give you a bit of orientation. This right here is your pubic symphysis. So this gubernaculum will be anterior to the pubic symphysis. Yeah, just to give you some orientation. So then as the testes descend, the gubernaculum gets a little shorter because it's bringing the testes closer to the exterior of the body. And then you have it where it eventually goes out of the body and into the scrotal sac. It is held in place by the gubernaculum. The spermatic cord includes, remember, all your vas deferens or ductus deferens, your nerves, your arteries, and your lymphatic vessels. Yeah. So that is the images of how the testes descend and eventually end up in the scrotum. So looking closer at the testes, you have all of these, this is the testes as a whole. You have all of these seminiferous tubules where you have the growth, the production and growth of the sperm. Once they are produced, they eventually make their way into the epididymis where they mature. And then they wait so that once it's time for ejaculation, they will leave the vas deferens and go, leave the epididymis, go through the vas deferens, and then meet up in the other parts of the body to be ejaculated out into the ejaculate, or with the with the ejaculate, they will leave the body into either the vagina or wherever he is ejaculated. So this is a microscopic view of this image. Make sure that you're looking at both so that you can get an understanding of how the sperm are made. Here you'll see that you have these spermatogonia. Spermatogonia are basically your very, very, very extremely immature sperm cells, and they eventually differentiate until they become mature sperm cells with the flagella. Right. And then once they do that, they sort of go into this epididymis because they're now in this lumen as mature sperm cells. They move into the epididymis, growing, creating a game plan and getting ready to go. So this is what that looks like on the inside. And again, this is a micrograph of that drawing. So you have your mature sperm cells with flagella in this lumen, getting ready to go into the epididymis. Sorry about that. Getting ready to go into the epididymis to be ready to be ejaculated. But here you have your spermatogonia that will eventually mature until they are fully mature with flagella. All right, this entire thing is a seminiferous tubule. This is a cross section of a seminiferous tubule. We talked about the and I still can't say it, the sustentacular cells. So go back, look at the word, look to see what these cells do, but these are here. All right, your sustentacular cells are here. Your basement membrane is the membrane that surrounds your seminiferous tubule. All right, basement membrane. And then you have these interstitial cells, also called the cells of Leydig. Interstitial cells, also called the cells of Leydig. Another micrograph of everything that we just talked, of what we just talked about. So you have, this is the vas deferens or ductus deferens. You have the sperm that are sitting in the lumen of that vessel. And then you have your smooth muscle. So remember, smooth muscle is a type of muscle, obviously, and some pseudocolumnar epithelium. You should be able to look at that and know what I mean when I say pseudostratified columnar epithelium, right? So go back to the first lectures in anatomy when we talked about tissues, the different types of tissues, to refresh yourself so that you can remember what pseudostratified columnar epithelium is. But that is what lines the lumen of the vas deferens. So now the penis. Here you have, in this cross section, you can see this is the corpus spongiosum, where the urethra is going through the corpus spongiosum. You have two corpus cavernosa one on either side of each other. And here you have your superficial dorsal vein and your deep dorsal vein. Remember, the deep dorsal vein was the one that took all the blood from the corpus cavernosa and drained that blood out so that the penis can go from erect to flaccid. We have the dorsal nerve, the dorsal artery, deep dorsal vein, etc. And again, the urethra goes through the corpus spongiosum. You have the prepuce, which is foreskin. You have the glans penis, which is the head of the penis. And then you have the external urethral opening where urine and semen will leave the penis. Urine and semen will leave the penis. All right. So make sure that you're looking at all of this tissue so that you can understand this is why it's important for you not to throw away the information that you've already learned, because it can help you understand other things. Everything builds on everything else. So we just talked about, let me go back. We just talked about these pseudostratified columnar epithelium. This was a lesson that we learned way back in the beginning of the first few weeks of anatomy and physiology. Here you have the skin of the penis. This is epithelial tissue. You have the subcutaneous layer. Remember, subcutaneous is not a part of the integumentary system, but it's often talked about with the integumentary system because it's very closely associated. You have your connective tissue or fascia. Then you have your arteries, veins, and nerves. You also have like different arteries, so deep artery, dorsal artery, etc. So make sure you're looking at all of that. This just goes through sexual stimulation, how it occurs. So you get stimulated. This is either by having a thought of something or someone, or your senses, right, being stimulated. So you can see something you like that you're attracted to, you can hear a sound that you like, you can smell a smell that you like, you can be touched in, in whatever ways, and that can stimulate you sexually. And then that will cause all of these changes, or it can cause these changes. So look through that so that you can know and understand how erection occurs. This chart just basically talks about sperm and semen characteristics in healthy males. So how much or the volume of ejaculate is important, because that will be a part of what determines the health and the virility of the male. The number of sperm, the concentration of sperm, the vitality of sperm, how much does it move, right? Or is it very active? Is it not very active at all? And they just kind of sit around like couch potatoes. That is very important. Or I should say how robust they are. The motility is how much they move. All right. And then the normal morphology, so how they look, because if they don't look the way healthy sperm will look, then they're not going to function properly because they don't have things where they're supposed to be. So this is what a sperm cell looks like. You have the head of the sperm, you have the mid piece of the sperm, and then you have the tail of the sperm. The tail obviously is the portion that allows it to move from in the vaginal vault into through the cervix, into the uterus, into the fallopian tubes, and eventually into the egg, if it's the lucky sperm. In the mid piece is where you have the majority of your mitochondria. Remember, the mitochondria is going to produce ATP for this sperm cell to actually live and do what it does. It takes a lot of ATP for it to do all of that. So that's why you have a lot of mitochondria. And then the head, you have the nucleus and the acrosome. So that this is where your genetic material is located, and this helps the sperm get into the egg Look this up so that you can know specifically what it does. Don't just depend on your PowerPoint. And this is just a micrograph of sperm cells so that you can actually see it. And I believe that is it. That is it. So that is the end of male reproduction, and we will start female reproductive system soon.